Dube from CSIR in Pretoria, South Africa, where he's a senior researcher. Ed Meyer is going to talk about nanomedicines for infectious diseases.
Okay, thank you, Mr. Chair, and the CLINAM organizers for giving me this opportunity to speak today. Basically, I'd like to talk about, change the gear a little bit and talk about infectious diseases and as we leave cancer. But I want to present about our approach in terms of infectious diseases, where we are now trying to move beyond just delivering the drug and having multiple functionalities from nanoparticles. So we think that this could be a future application in addressing infectious diseases. So just to present the landscape in terms of the burden of infectious diseases, we have three main killers in infectious diseases: HIV, tuberculosis, and malaria. And there are a lot of deaths. I won't go so much into the statistics, but you have about 1.7 million deaths from HIV, 1.4 million deaths from tuberculosis. And the burden, as you can see from the coloring, the reds and the dark colors, you see that the burden is mostly in the developing countries. But while that is also the case for the developing countries, you do see the spread of these diseases outside of developing countries into Europe and the Americas and other developed countries. So it's just not a problem for Africa and the developing countries. So we all have to consider infectious diseases. So with that in mind, in terms of the spread of the disease, the problem that we have is we have increasing and emergence of resistance of the infectious disease pathogens to existing drugs. But while those drugs that we have are starting to not work, we have a very poor pipeline of incoming new drugs that are being developed. So it's kind of a dire situation that we're faced with. So that is now the landscape that we see when we were trying to develop these nanomedicines, is how can we improve the current drugs that we have, because we don't have any really new drugs to work with. And at the same time, how can we apply these medicines in a way that we also overcome the resistance that we see? So that is where the multifunctionality of the nanoparticles that we are trying to develop comes in. So in the talk that I'm presenting, I'm just going to talk about the approach that we have. It's still early days. We're still at an in vitro stage. We haven't done infected animal models yet. So I'm just going to focus on tuberculosis. So as we design our nanoparticle system, the important thing for us is to understand how the disease pathology functions, so that we can then identify the target that we need to aim with our nanosystem. So when it comes to tuberculosis, the pathogen is residing within macrophages. So those are the cells which engulf the bacteria, the pathogen, when you get infected. And in most of the life cycle of the pathogen, you see the bacteria within the macrophage. So that is now our target in terms of our nanoparticle design. But when we look at the events that are happening within the pathogen, within the macrophage, for some reason the pathogen is able to survive within that macrophage. And three key aspects which the pathogen uses to survive within the macrophage are related to it suppressing the immune response, the macrophage immune response to the pathogen. For example, it can switch off secretion of pro-inflammatory cytokines or reactive oxygen species and nitrogen species, and prevents the maturation of phagosomes. So then this now brings an immunomodulatory aspect to our approach. So for example, in the last talk, you're talking about delivering cytokines. But in this case, now we're thinking of stimulating the macrophage in order to tell the macrophage that there's something foreign inside you and you need to destroy this pathogen. So we are now delivering immune-stimulating ligands to macrophage surfaces. We stimulate the macrophage in order to be able to destruct the intracellular pathogens. And at the same time, we can also deliver our conventional drug compounds into these macrophages. So now we have a two-pronged approach to eradicating the bacteria, the immune modulatory and the drug delivery. So we went about to identify a ligand on the surface of the pointer should be. Oops. Okay, how do I get back to? Okay, to identify a ligand, or sorry, a surface receptor, which eventually with downstream processing, we do get the immune properties that we're looking for. That is reactive oxygen species burst, our pro-inflammatory cytokine release, and the like. So the receptor we're working with is dectin-1, and then we've now conjugated to a PLGA polymer backbone, a polymer with immunomodulatory properties, which is specific for dectin-1. So that is what you have at the top left there. So we have our immune modulating polymer. We chemically conjugate that to PLGA, and then now we have our biointegrated copolymer that we synthesize. And from that polymer, we can then synthesize the nanoparticles and encapsulate our anti-TB drugs. So now we have a biointegrated multifunctional nanoparticle. So we can control the density of our immune modulating ligand on the surface of the nanoparticles, and we anticipate that this can then modulate the immune response that we get from the particles and also the pharmacokinetics of the drugs inside the cells. If we incubate those particles with human macrophages, you see the DAPI is the stain of the nucleus, and the green is the fluorescence from your particles. We do get uptake of those particles. If we do some flow cytometry, you can see a shift, increasing granularity, so we get particles being taken up. And this is a scanning electron microscope of the particles. They're about 300 nanometers in size. And our drug loading, we can put about 1% weight by weight of our drug into those particles. So here we can confirm with the red stain there that there's the presence of our dectin-1 receptor on the macrophages that we're working with. And if we incubate our nanoparticles with those same cells, so the green there is your nanoparticles, you will see that the particles do interact at the surface of the particles. And then we've also done some Luminex in order to quantify now pro-inflammatory cytokines which are being produced by these cells. And we can do the study over 2 hours and 24 hours. And especially at 24 hours, we do see a difference in the concentrations of tumor necrosis factor alpha, interferon gamma, and interleukin-12, which are your pro-inflammatory cytokines. We have a significant difference between particles which do not have our immune modulating ligand and particles which have the ligand. So you get a boost in an increase in your pro-inflammatory cytokines. In the absence of this ligand, you still do see some stimulation of these pro-inflammatory cytokines. And this comes about as just normal phagocytosis of particles. But when you have that immune modulating ligand, you can increase the response that you get. And we are going to play around with the density of the ligand to see if you can tune or control that response that you get. Again, looking at reactive oxygen species, we see a similar pattern where we can boost up the ROS produced by the cells. And we've done some cytotoxicity studies, MTT assays looking at whether these particles are toxic to these cells because of this ROS and RNS, and we do not see a difference with controls. Okay. And again, the second aspect, as I mentioned, is now the intracellular drug delivery. We're doing some intracellular pharmacokinetics to see how these particles are able to deliver our TB drugs into the cells. And we see an enhancement, which is what we expect in terms of concentrations of our TB drug, in this case rifampicin, where you have a rifampicin control, and then you compare it to particles without your ligand and particles with the ligand. And if you calculate the area under the curve, we find that the particle with the ligand has a much higher intracellular concentration of your TB drugs. Stimulating or activating the dectin-1 also enhances cell phagocytosis beyond that which you get with normal particles which do not have the ligand. Okay. And if we do the same experiment, those graphs are THP1 cells, and then we have the human macrophages, and we also see a similar pattern where after 24 hours, after 2 hours, you do get a significant enhancement in intracellular drug concentrations. So in summary, like I said, these are still early days, and we're going to do a bit more in terms of proving this concept in infected cells as well as in animals. But I just wanted to point out that we're working on what we call biointegrated nanoparticles, where we can have multifunctionalities from the cells, and we can increase the response of the cells to intracellular pathogens, and at the same time deliver the drug. And then now we want to show whether we can tune the immune response that we get by playing around with our ligand densities, and then we can see the impact on infected macrophages and animals. So I'd like to thank you for your kind attention and acknowledge the work that we've been doing here at the CSIR in South Africa, and also with the University of Buffalo in the US, where some of this work was done. And if you have more interest in it, we have a publication on that in the journal. The citation is at the bottom. Thank you.
Thank you. Thank you very much. Are there any questions? Yes, in the back.
Thank you very much. I have actually two questions. The first one, how would you expect your nanoparticles be specific for lung macrophages? How would you then, because I mean, if you stimulate the general macrophages within the body, I mean, this could be quite dangerous.
So with the current design, they're not specific. They're not specific for infected macrophages. They're specific for macrophages in general.
Yeah.
And we're thinking that we could have this formulation to be inhaled, because TB is a disease of the lungs. So if you inhale these particles, they're going to be taken up by general macrophages in the lungs, which are the macrophages which are infected in any case. So, and the cells, I mean, the particle PK should be such that they will be cleared within that area, and you don't get a lot of the distribution of the particles outside of the lung tissue and into the bloodstream.
Mm-hmm. But the part would still enter the...
Sorry?
The part of would still enter the bloodstream, okay, but I understood. Also through the lung. It would enter bloodstream anyway, so...
Yes, but probably small amounts of particles would cross the epithelial membrane into the bloodstream.
Mm-hmm. But would it be also specific for monocytes then? I mean, if it comes to the blood, would monocytes have the same
so the
ligand?
Dectin, so yeah, probably it would, but the dectin receptor is highly concentrated in alveolar macrophages, and we don't see a lot of it in the other macrophages. But there is a chance that we will see some non-specific uptake in the other cells.
Because then you activate monocytes and in blood, if it is. And the second, maybe just a short comment, would it not be also kind of interest for the cancer, like stimulating macrophages, which are also kind of suppressed in the tumor, maybe?
Yes, yeah, that'll certainly, I think there's a potential application in that area as well. But our focus is mostly on the infectious diseases, where we have a bit more experience.
Yeah, thank you.
Yeah.
Any more questions?
Yes, regarding your strategy, the macrophages in the lung are specialized just in not being activated because it is very dangerous for the alveoli. How do you think your strategy is going to impact in the working of the lung? Because if you induce an immune reaction, an inflammatory reaction in order to avoid the infection, what would be the outcome for the alveoli?
So that's why we want to see how we can play around with the ligand densities in terms of controlling the immune response we get. And we're also looking at anti-inflammatory. Here I just presented the pro-inflammatory cytokines. So we will see whether we can get a balance between the pro and the anti-inflammatory and see how that works. But like I said, this is still early days, and we still have quite a bit of a way to go in terms of understanding it and trying it out in animal models. But I agree with you, we'll need to see a balance between pro and anti-inflammatory cytokines in order to not get a chronic inflammatory state.
More questions? Yeah, I have a question. you're focusing on infectious disease. Which is the first? What is your order in terms of priority? Where are you going to apply it first?
Tuberculosis is one of them. Malaria, there's a potential for malaria, liver stage malaria targeting in that aspect. And from some of the discussions, I think even diseases like Chagas disease, where you have parasites within macrophages, is another potential area. But we'll start with tuberculosis.
Thank you very much.